Hi, everyone. So let's talk about blood. Blood is a connective tissue. So why, you ask? Well, because it is cells in a non-living extracellular matrix. The cellular components are referred to as formed elements. Some of these cells are complete cells with nuclei, but not all. The matrix is referred to as plasma. Plasma contains water, solutes, plasma proteins, pretty much anything that's transported in this medium. And we can divide these formed elements into three groups. Platelets, which are also called thrombocytes. These formed elements are really pieces of a larger cell. So as a result, they consist of a plasma membrane, cytoplasm, and cytoplasmic granules, but that's it. No organelles. Leukocytes, formerly known as white blood cells. These formed elements contain a full complement of organelles and are often distinguished from each other in part by their distinct nuclei. These are the only true cells of the blood. And erythrocytes, formerly known as red blood cells, human erythrocytes are anucleate, which means no nuclei. They're biconcave in shape, which increases their surface area and makes them extra flexible. Most of the formed elements in human blood are erythrocytes. In this stained smear, you can see the erythrocytes and the large, distinct nuclei of the scattered leukocytes. They're the purple ones. Okay, here we see those biconcave erythrocytes. Again, that shape increases surface area and flexibility. The function of carrying respiratory gases will be discussed in detail in our lecture content. Lastly, erythrocytes stack one on top of the other in columns called rouleaux. They'll travel through capillary beds in this arrangement. That biconcave shape assists with the formation of those rouleaux. Leukocytes can be divided into two categories. These cells are considered granulocytes because they create and maintain granules in their cytoplasm. The cytoplasmic granules take on different stains and so appear as different colors. In addition, the nuclei have different shapes. So we can use these two things, color of cytoplasmic granules and shapes of nuclei, to identify them. Neutrophils are referred to as being polymorphonuclear, which means many-shaped nucleus. This is a great description because their nuclei have many lobes. Think of one house with many rooms. That's kind of how their nuclei work. As a result, they often appear as having several nuclei. I like to think that they look like the planet Earth or a smiley face. These cells have stained cytoplasmic granules. However, the granules pick up just a little bit of purple stain, so they appear light purple or lilac in color. Those granules contain enzymes and defensins, all of which assist in their function. So these cells phagocytize bacteria in the short term. We're going to learn their fate in the immune system lecture content later. Now, eosinophils contain reddish granules and a U-shaped nucleus. Really, those granules are a key identification characteristic. They're involved in allergic responses, but are really good at killing microscopic parasitic worms. Again, we'll learn exactly how they do that in the immune system lecture content. The last granulocyte of our lab are the basophils. Their granules are large and pick up a lot of purple stain, so they appear dark purple. You can barely see that bent nucleus in there. These cells release those granules which contain histamine and heparin, and that stimulates inflammation for tissue repair. Now, leukocytes that do not have cytoplasmic granules are referred to as agranulocytes, and there are two on our list. Lymphocytes have a dark round nucleus that's large. So it's surrounded by a thin band of lighter staining cytoplasm. For now, there are different types such as T and B lymphocytes. T and B lymphocytes, T lymphocytes conduct your immune response. There are a bunch of different T cells. So more about that later. B cells secrete antibodies with neutralize antigens or pathogens. Monocytes have bent nuclei, so either C or U-shaped. These cells wander through the body. Sometimes they'll stop in an organ and live there. When that's the case, we call them macrophage. And by the way, you've met some of these in AMP1. The dendritic cell of the skin and microglia of the brain are a type of macrophage. So these cells phagocytize bacteria and lyse them into small pieces. Some of those pieces are attached to self-surface receptors in an event called presenting the antigen. And you guessed it, we'll learn more about that in immune system content later. Now, not only do we want you to accurately identify each formed element and explain their function, but we also want you to know their numbers. So this is a reminder for me to tell you that you should never let monkeys eat bananas. That's going to make sense in a little bit. Okay. We can stain a patient's blood sample with that right stain and get all those formed elements to show us their characteristics. We can then count up the numbers of each formed element on a grid and calculate the percent of each. On this chart, we've already counted the numbers for you of each of those cell types on that grid. And I'm showing you this data. When asked to calculate the percentage, here's what you do. First, add up the total. Then, divide the number of the indicated cell by that total and multiply by 100. But don't stop there. In the lab manual is a chart showing you normal percentage ranges. That percentage range of normal must be in your head during a lab exam. The way our leukocytes work is that this is the normal circulating percentage of each. If you have an immune reaction, for example, a parasitic worm, you'll make more of the leukocyte that you need to get rid of that infection. So if you have microscopic parasitic worms as an infection, your red bone marrow will make more eosinophils, and your percent of them will be higher than this normal range. So here's an example of a calculation there. The percent of monocytes would be the number of monocytes, here 12, divided by the total number of leukocytes here, which is 174. And then you'd multiply that by 100 to get the percent. So what did I mean by never let monkeys eat bananas? Well, never let monkeys eat bananas is a mnemonic that reminds us of the most numerous to least numerous leukocytes. Never equals neutrophils, let equals lymphocytes, monkeys equals monocytes, eat equals eosinophils, and bananas equals basophils. So I think you should take a few minutes to practice identifying these formed elements. I'm not going to give you the answer. And so here are your hints though. Ready? Erythrocytes are biconcave. Platelets are tiny purple dots. Neutrophils have a multi-lobed nucleus. Eosinophils have red granules. Basophils have large purple granules. Lymphocytes have a round nucleus and no granules. And monocytes have a bent nucleus and no granules.